morning. Welcome back to another online lecture. So welcome back to online lecture number 2, the day 3 of aerobic respiration. So I hope you all are having a wonderful day and everyone is staying safe. So what we are going to do in this lecture is we are going to talk about stages 3 and 4 of aerobic respiration. We're going to give a general overview of the stage, and then we're going to talk about what goes in and what comes out. And again, all of this is part of our overarching goal of answering the question of how does a cell break down glucose to obtain energy? So in our journey of aerobic respiration, we are here on stage 3. So far what we have covered is in stage 1, or glycolysis. We take glucose, which is a 6-carbon molecule, that is broken down to a 3-carbon molecule pyruvate. In stage 2, we take this pyruvate, and using pyruvate dehydrogenase, then we lose 1 carbon as CO2, and the acetyl group, or that 2 carbons that is left of pyruvate, becomes acetyl-CoA. So now we're going to answer the question of, all right, what happens next? What do we do with that acetyl-CoA, and what is happening in the citric acid cycle? So before we get really deep into the citric acid cycle, I want to first tell you a little bit about some generalities of what happens, and then I also want to tell you the other names that it goes by. So just like many things in biology, there are several names for the citric acid cycle. For the citric acid cycle, you can also call it the TCA cycle. So TCA stands for tricarboxylic acid. And as if 2 names wasn't enough for a cycle, this cycle can also go by the Krebs cycle, and that is named after Hans Krebs, which is the scientist that identified all of the components of the citric acid cycle in 1937. So where is this happening? Where are we? That's going to depend on whether we're talking about a eukaryotic cell or a bacterial cell. So for eukaryotes, we are in the mitochondrial matrix. For a bacteria, we're going to be in the cytosol. So for them, they don't have to worry as much about moving things around like eukaryotes do. So let me give you a brief overview of what happens in this cycle. So in this cycle, we are going to start by taking the acetyl group, and this acetyl group is coming from acetyl-CoA, and this acetyl group is what is left of glucose, and we combine that acetyl group with a 4-carbon molecule, and that 4-carbon molecule that we combine it with is oxaloacetate. And this is going to make citrate, and the citrate is then used in the cycle. And we're going to kind of leave our general overview at that. We will actually draw this process out, and you'll see some of the other details of what occurs. So the last thing that I want to leave you with on this page is this cycle is so important that no deficiencies exist. And I started spelling deficiencies way wrong. So no deficiencies exist. If during development a deficiency does exist, then that embryo is not viable. And so you might be asking yourself, why is this the case? Why is this citric acid cycle so important that we can't have any mistakes in it? And to answer that question, we actually have to look a little more broadly and look at the metabolism of various macromolecules. So you and I know from Unit 1 that there are more than just sugars in terms of macromolecules. We have 4 macromolecules, and this chart does a really nice job of showing us, at least briefly, the metabolism of these various macromolecules. So you and I have been working right here. So we've talked about sugars going through glycolysis, becoming pyruvate, then acetyl-CoA, and now we're talking about how that acetyl-CoA goes into the Krebs cycle. So let's take a look at our lipids and fats. We have fatty acids that go through something known as beta oxidation, and if you're interested in all the nitty-gritty details of what happens in metabolism in this chart, you'll actually learn it in biochemistry. So after it goes through beta oxidation, we have acetyl-CoA that gets fed into the Krebs cycle. If we take a look at amino acids, they go through a process known as deamination, and that gets fed into the Krebs cycle. We take a look at nucleotides. Guess what? They also get fed into the Krebs cycle. And so regardless of the type of macromolecule that you are, every single macromolecule throughout its metabolism has to be fed into the Krebs cycle. And so if you have a deficiency, a mutation, a problem in that Krebs cycle, then you can't break down any sort of macromolecule, which means you can't harness any energy. And so that is why the citric acid cycle is so important. In the case of pyruvate dehydrogenase deficiency that we talked about, that only affects this step right here. And so if you have a mutation or a problem in this step, you are still able to go through some of these other mechanisms of breaking down macromolecules. And so again, Krebs cycle, the citric acid cycle is important for every single type of metabolism for all macromolecules. Therefore, no deficiencies can exist. Okay, so now that we've covered, and hopefully I have convinced you that this cycle is in fact really important, we're going to talk about a new energy management molecule. So far, the energy management molecules that we have been talking about are ATP and NAD+ / NADH. So those are ones that we are familiar with. Well, it turns out that in the citric acid cycle, we actually have a new energy management molecule, and that is FAD / FADH2. And this FAD stands for flavin adenine dinucleotide. So let's go ahead and draw the energy reaction coordinate diagrams for these. It'll be good practice. So I'm going to write up my little cheat sheet here. So our FAD is going to be oxidized, and our FADH2, which has gained 2 hydrogens, it has gained electrons, is the reduced form. All right, so I'm going to go ahead and draw 2 reaction coordinate diagrams. So let's start with, actually, let me be good first and label my axes. As you all know, I'm horrible about labeling my axes for these. I kind of get in the groove and forget about them. So let's start with FADH2 going to FAD plus 2H. So it is losing electrons, so it is oxidized. So when we get the oxidation of FADH2, this delta G is going to be less than 0 or negative, so this is a favorable reaction, and so this is going to release energy. Now, when we are taking FAD and adding 2 H's or adding 2 electrons, we're gaining electrons and we are reducing. So when we get the reduced form of FAD, we end up with an endergonic reaction. And so delta G is greater than 0, and that requires an input of energy. So we will see this new energy management molecule as we go through the citric acid cycle. Okay, so now that we have a general idea of what's going on, we're going to take that acetyl group from acetyl-CoA and combine it with citrate. Now we are actually ready for the citric acid cycle. So we're going to start with a blank page, and we're going to draw a simple model of what's going on. So we're going to start with our 4-carbon molecule, and this is that oxaloacetate. That we said we are going to combine acetyl-CoA with to make citrate. So let's go ahead, and I'm going to use different colors. So here is our acetyl-CoA. That CoA is removed, and we take that acetyl group, and that acetyl group gets added onto that oxaloacetate, and this is what forms citrate, which is a 6-carbon molecule. And I'm trying to keep things color-coded here. So the dark blue are the carbons that come from originally from glucose, and the light blue are the carbons that are from the citric acid cycle. So after we make citrate, we're going to take that citrate, and we're actually going to make a 5-carbon molecule. And just for sake of time and for simplicity, we're just going to call it a 5-carbon molecule. We're not even going to give it a name. While this happens, we are going to do 2 things. One thing is, you'll notice that we lost one of our dark blue carbons, and that is because one of those carbons leaves as CO2. The other thing that happens during this step is we take NAD plus and we reduce it to NADH. So far we have made 1 NADH, and we have lost 1 carbon as CO2. We are then going to take that 5-carbon molecule, and now we're going to make a 4-carbon molecule. You'll notice that now we have no dark blue, and that is because during this step we have lost another CO2. In addition, while we have been knocking off one of those carbons, the last carbon that is left from our pyruvate, we are also taking another NAD plus and reducing it to NADH. So now we have this 4-carbon molecule, and we want this 4-carbon molecule to be rearranged back to oxaloacetate so that we can repeat this cycle. And that's going to go through a series of steps. And while those steps are taking place, a couple things happen. First, we take an NAD plus and we reduce it so we get another NADH. And then here's where that new energy management molecule comes in. So we take an FAD, and that is reduced to FADH2. And then lucky for us, in addition to that, we also take an ADP and we make an ATP. So again, in the citric acid cycle, we start with this 4-carbon oxaloacetate. We take that acetyl group from acetyl-CoA and we combine those to make citrate. We then take this citrate 6-carbon molecule. We knock off a carbon that leaves this carbon dioxide, and now we're left with a 5-carbon molecule. Because that is an exergonic reaction, we can couple it with an endergonic reaction, which is right here, the formation of NADH. We repeat that process by taking a 5-carbon molecule, knocking off that last carbon from pyruvate to make a 4-carbon molecule, and with that we can reduce NAD plus to NADH. We lose that carbon as CO2, and then finally this 4-carbon molecule is rearranged back to oxaloacetate, and during that step we reduce another molecule of NAD plus to NADH, and we reduce a molecule of FAD to FADH2, and we make an ATP. So at each of these steps, one thing to keep in mind is that enzymes are playing a role. So just like in glycolysis, where there was an enzyme at every single step, there's an enzyme at every single step of the Krebs cycle as well. So one thing that we have to think about is, well, how many times do we actually have to complete this cycle? We have only drawn one cycle, and we have said that this can continue. Well, we have 2 acetyl-CoA molecules because we had 2 pyruvates made. So one thing to keep in mind is that even though we've only drawn one cycle, we actually have to complete this cycle 2 times because we have 2 acetyl-CoA molecules. So let's take a look at what a more complex model looks like. So again, I don't expect you to have this memorized. We are focusing on what goes in and what goes out, but I do want to show you a bit of a more complex model. So here at the top, we're starting with that oxaloacetate. Here's our acetyl-CoA. You'll notice that we have citrate synthase. So that's the first enzyme. That is what makes citrate. And then we are taking that citrate. It goes through a series of steps. And throughout those series of steps, here's our first CO2 that we lose. Here's our second CO2. So now we've lost the 2 carbons that are left from pyruvate. You'll notice that we have our 2 NADH molecules that are formed, our FADH2, and then another NADH molecule up here, and then our ATP that is formed. So again, I don't want you focused on memorizing these steps. I just want you to have an overview of what goes in, what comes out, and where are those carbons from glucose coming. So if we wanted to draw our energy reaction diagrams, so here is our G and our time. At the start of the citric acid cycle, we begin with acetyl-CoA, and then we end with 2 CO2s. And so that is our exergonic reaction. And anytime we have an exergonic reaction, we have energy that is released. We want to harness that energy, and the way that we harness that energy is by putting it into the bonds of our energy management molecules. And so we get a series of endergonic reactions that can occur. So we can take ADP plus P to make ATP. We can take our NAD plus, that is reduced to NADH. And then we also have FAD that is reduced to FADH2. So again, all 3 of those endergonic reactions would look the same, which is why I'm using one diagram to model them. So again, in the citric acid cycle, just like in stages 1 and 2, we couple endergonic and exergonic reactions together. So now that we have walked through the citric acid cycle, let's do some practice problems. And the first practice problem that we are going to do is answering the question of how many steps in the citric acid cycle involve oxidation of a metabolic intermediate. So again, I'm going to encourage you to go ahead and pause the video and to work through this problem on your own. And then when you come back, I will be here and we will walk through this together. So hopefully you have gone ahead and paused the video and attempted the problem on your own. So for this problem, we are looking at the oxidation. So whenever you see oxidation, you should automatically be thinking about redox reactions. And so this question is really asking us, how many redox reactions are there that happen? So let's go ahead and write up our cheat sheet. So Leo Gur. And if we are looking at redox reactions, we're looking at the loss or the gain of electrons. And in my mind, because we are biologists, we're looking at loss or gain of hydrogens. So let's take a look at, and you can use either your simple model of the citric acid cycle that we drew or this complicated one. Let's look to see if we can find places where we have either a loss or a gain of hydrogens. So the first thing that jumps out to me are these NAD plus molecules that are reduced to NADH. So they are gaining hydrogens or gaining electrons. We also have a molecule of FAD that is reduced to FADH2. So in the citric acid cycle, if you count the things that I have circled, we have 4 steps. And so anytime you have a reduction, because redox reactions come in pairs, you also have an oxidation. So we identified the reductions, which tells us where the oxidations are. So 4 steps in the citric acid cycle are going to involve oxidation of a metabolic intermediate. So let's take a look at problem number 2. So problem number 2 states, each glucose molecule that enters cellular respiration and is catabolized by glycolysis and pyruvate oxidation results in the production of X-NADH and X-FADH2 in the citric acid cycle. So this problem is asking us to tell us how many NADH molecules are produced and how many FADH2 molecules are produced in the citric acid cycle during glycolysis. So let's take a look at our citric acid cycle that we have here on the left. So we have 1, 2, 3 NADH, and we have 1 FADH2. But this is for 1 molecule of acetyl-CoA. So we know that there are actually 2 molecules of acetyl-CoA because at the end of stage 1, we produced 2 pyruvates. Each of those pyruvates was then converted to acetyl-CoA. So we actually have 6 NADH that are produced for 1 glucose, and we have 2 FADH2 molecules that are produced per glucose. So again, the key here is each glucose. So a really good way to test yourself on this type of problem, or a really good application of this, would be if I was to ask you, well, if you have 2 glucose molecules that are going through the citric acid cycle, how many NADH would you get? Or how many FADH2 would you get? So again, this is that hint that I gave you that we have to keep in mind that we need 2 turns of the citric acid cycle per glucose molecule. All right, I hope you all are ready for practice problem number 3. So for practice problem number 3, this is asking us for each glucose molecule that is catabolized by glycolysis and pyruvate dehydrogenase, X carbon atoms enter the cycle as acetyl-CoA and X carbon atoms leave the cycle as CO2. So again, we're going to go ahead and we're going to use our chart that we have, and I'm going to circle the CO2s. So those are our CO2s that leave. So we get 2 CO2s that are leaving. And then up at the top here, this is our acetyl-CoA, and our acetyl group has 2 carbons. So we have 2 carbons coming in from our acetyl group and 2 carbons leaving as CO2. But just like the previous problem, this problem is asking about each glucose. It's not asking about pyruvate or acetyl-CoA. It's asking about the glucose molecule as a whole. So the glucose was split into 2 pyruvate. Each pyruvate was converted into acetyl-CoA, and then each of those acetyl-CoAs went through the citric acid cycle. So we actually have to multiply this by 2. So we get 4 carbon molecules that entered the cycle as acetyl-CoA and 4 CO2 carbon molecules that leave. So let's go ahead and fill those in. So hopefully you all are with me. Again, the key here is to remember that the citric acid cycle, we have to go through it 2 times per glucose molecule. If you have any questions please let me know. I can go through this with you one-on-one as well. Feel free to contact me in any of the ways that are listed in our syllabus. Okay, so let's go ahead and summarize everything that we have said so far about the citric acid cycle. So this is the third stage of aerobic respiration. For eukaryotes, this is happening in the mitochondrial matrix. For bacteria, this is occurring in the cytosol. In terms of what goes in, we have 2-acetyl-CoA. What comes out, we have 4 CO2, 6 NADH, 2 FADH2, and 2 ATP. If we were to summarize what happens in the citric acid cycle in one sentence, we can say that the acetyl group from acetyl-CoA enters the cycle and leaves as CO2, producing several energy management molecules. And those are listed above. So that NADH, FADH2, and ATP. Okay, so now that we've gone through a couple of stages, it's really important for us to not only summarize each stage, but to do a summary of our energy management molecules that we've gained so far. So in glycolysis, do you remember how many ATP we gained? Hopefully you're yelling at the screen and you were telling me 2 net ATP. So we made 4 ATP, but we had to invest 2. So we have 2 net ATP, 2 NADH. For the production of acetyl-CoA, we actually didn't make any ATP, but we did make 2 NADH. In the citric acid cycle, we made 2 ATP, 6 NADH, and then this is where we introduce that new energy management molecule. So we made 2 FADH2. So far, our running total is 4 ATP, 10 NADH, and 2 FADH2. So not too shabby. Okay, so before we move on, you'll notice that there's a link on this page on the top of it, and I will actually post this link in the description box as well, but I did want to go ahead and play at least a little bit of the link for you.
Of the mitochondria of all the cells is the cycle of reactions that will hand Krebs a Nobel Prize. The cycle takes the energy and food and makes it into other forms your cells can use. Krebs cycle makes electron carrier NADH, which later brings electrons to the electron transport chain. And Krebs makes FADH2, its function is the same. Krebs also makes some ATP, another claim to fame. We're talking Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2. Right before the cycles are...
All right, so I'm going to go ahead and pause it there. He does a really nice job of continuing throughout the rest of the song, and he actually goes through each of the steps in depth of the TCA cycle. So one day when you take organic chemistry and you need to memorize all the structures and all of the enzymes at each of the stages, it's a great song because he does a nice job of having a really catchy tune. So I actually will find that my husband randomly sings this song. He's the person who actually introduced me to this, and every now and then he'll just, start singing It's the Citric Acid Cycle Krebs. So it's kind of a running joke in our household, but I wanted to share that with you because it makes me smile every time that I hear it. So although we have only covered stages 1 through 3, we are actually ready to go back to this question and answer it. So this is the question that before break, so 2 weeks ago, that I had asked you all when we started aerobic respiration. And we actually had students that answered A, B, C, D, and E, so all over the place. So now that we have gone through the majority of aerobic respiration, you can actually answer this question. And so this question was asking you about the idea of what happens when you work out. Where does the carbon from the fat go? And first, if I wanted to update this question, I've seen a lot on Twitter, a lot of jokes. Instead of talking about freshman 15, the quarantine 15. So this idea of, when we are working out, where does the food that we are eating, where does that carbon from the fat go? So hopefully you are telling me, all right, Dr. Hubbs, we get it. We know what the answer is. And you're yelling at the screen and you're telling me that it is E. So during stage 2 and stage 3, we see the production of CO2. And that CO2, that carbon, comes originally from glucose. All right, so I just quickly wanted to go back to that question and make sure that we are all on the same page. So now we are finally at stage 4. So for stage 4, we are going to talk about electron transport and chemiosmosis. So let's start with some general properties of what's going on in this stage. So the first thing that we're actually going to talk about is where this occurs. If you are a eukaryotic cell, this is going to happen in the inner mitochondrial membrane. For a bacteria, this is going to happen in the plasma membrane. Of course, bacterial cells don't have mitochondria, so no way that it could happen in a mitochondria. So to understand what happens in this stage, we actually have to look at the name. So this stage goes by 2 different names. It can go by electron transport and chemiosmosis. So that's kind of the first name. And the second name, which is also listed on this page, is oxidative phosphorylation. So to understand what's going on, we can actually take a look at the names. So we're going to focus on the left side first. So when we think about electron transport and oxidation, what this really means and what this is really referring to is NADH and FADH2. So those were our molecules that were reduced and have been reduced throughout stages 1 and 3. They are going to be oxidized. And when something is oxidized, they lose electrons. And so then their electrons are passed to an electron acceptor. In aerobic respiration, this is oxygen. And when you pass an electron and an electron acceptor is gaining a receptor, that's going to be reduction. So we actually have oxidation and reduction that is occurring. And again, in aerobic respiration, that final electron acceptor is oxygen. So the idea of electron transport and oxidative comes from NADH and FADH2 being oxidized. We take those electrons because they're essentially from stages 1 and 3, they're carrying those electrons so we can use them in stage 4. So they're electron carriers. So we take those electrons and we pass them through an electron transport chain, and then we pass them on to a final electron acceptor in the form of a reduction step, which in the case of aerobic respiration is oxygen. Now let's take a look at the right side. So we have this idea of chemiosmosis and phosphorylation. So let's go ahead and actually define chemiosmosis. And chemiosmosis is actually something that we have already seen in this class. We just haven't put a word to it. And this is just the idea of ions moving down their concentration gradient. So again, we've already seen chemiosmosis, we just haven't defined or given it a specific word. So in chemiosmosis, or the phosphorylation part of oxidative phosphorylation, what we have is we have protons that are pumped during electron transport. So that's during that oxidative step. And then when the protons go down their concentration gradient, they power the synthesis of ATP. So again, when we talked about things moving down their concentration gradient, we talked about the idea that a concentration gradient is a form of potential energy. And so in the case of things like cotransport, we can use something that's going down its concentration gradient to power an endergonic reaction, which is moving something against the concentration gradient. So here what we're doing is we are pumping protons. Those protons make a concentration gradient. When those protons go down their concentration gradient, which is an exergonic reaction, we couple it to an endergonic reaction. So hopefully you all are ready. I recommend grabbing a bunch of different colors because I'm going to do my best to color code everything as best as I can because there's going to be a lot of moving pieces. So let's go ahead and start with a blank page. And we're going to start first by drawing a really tiny little mitochondria up here in the corner. So to remind ourselves of where we are actually zooming in on that inner membrane. So we're going to just draw part of that inner membrane. We're not going to draw the entire mitochondria. We'd be here for forever and a day. And what we're going to do is we're going to draw 5 protein complexes. So just like in everything else where enzymes are completing all of the steps, enzymes are completing the steps in the electron transport chain and in chemiosmosis. So we're going to go ahead and draw, this will be complex 1, complex 2. These complexes do technically have names. We're just going to call them complex 1, 2, 3, so on and so forth. Here is complex 4. We will give complex 5 a name. Complex 5 is ATP synthase. So you can probably already guess what that complex does. So synthase, so synthesizing ATP. Oop, I did not mean to erase that. Let's see. Okay, so let's go ahead and draw our membrane. So now we've got our 5 complexes. We're going to label just to orient ourselves. So this is the inner mitochondrial membrane. This is the inner membrane space. And on the inside we have the matrix. So one thing that is going on in the matrix that we just finished talking about is the citric acid cycle. So let's go ahead and actually draw citric acid cycle. And we're not going to draw the whole cycle. We're just going to draw a simplified version of it. So we're going to draw our CO2 coming off of it. We know that NADH is produced in the citric acid cycle, and the other molecule that we reduce is FADH2. Okay. I'll actually move this down a little bit. Perfect. Okay, so far what we have is we have these 5 enzymes, these 5 complexes. We just finished talking about the citric acid cycle, and now what we're going to do is we're going to talk about what the role of each of these complexes is and what happens. So on the top, starting with complex 1, going to complex 4, this is the electron transport chain. And as we draw and fill out more of our diagram here, more of our model, you will learn why that is the case. On the right, this is the part that is chemiosmosis. So let's just put our labels up, and then as we draw the process, you will hopefully see where those pieces come from. So let's start with our NADH. So we said that in the electron transport chain, or in the oxidative part of oxidative phosphorylation, NADH and FADH2 are taken and they're oxidized. So we're going to take this NADH and we're going to make NAD+. Those electrons that are taken from the NAD, we also get an H+, so we take those electrons, which is 2 electrons, and those go into the electron transport chain. So they're actually going to be passed along down the complexes. So while this is happening, while we're taking that NADH and it is being oxidized, we are also taking protons and pumping them. So the role of complex 1 is to not only oxidize NADH to NAD+, but it takes an electron from NAD, which is why we have NAD+, and an electron from H, which is where the H+ comes from. Those electrons are taken and are shuffled into the electron transport chain, and then the protons, using that energy, because again, going from NADH to NAD+ is an exergonic reaction. Using that energy, we're then able to pump protons, which is an endergonic reaction. So now we're going to repeat the same process, but we're going to do it with FADH2. So complex 2 takes FADH2. It's going to oxidize that to FAD. It's going to then make 2 protons as well, and we're going to take those 2 electrons and they're going to be shuffled into the electron transport chain. So as we continue on, those electrons are going to be continued to be passed through the electron chain until they get to complex 4, and we'll deal with complex 4 here in a second. Complex 3, just like complex 1, is also pumping protons from the matrix to the inner membrane space. Complex 4, just like complexes 1 and 3, is also pumping protons into the inner membrane space. So complexes 1, 3, and 4 pump protons into the inner membrane space. Complex 1 oxidizes NADH. Complex 2 oxidizes FADH2, and taking those electrons from that oxidation is what is helping to power the pumping of the protons into the inner membrane space. So now finally, complex 4, we have reached the final electron acceptor. So at this stage, we take the electrons that have been passed through complexes 1 through 4 in a series of redox reactions, and they are finally accepted by a final electron acceptor, and that is oxygen. So we're going to take half of an O2, which is kind of weird to write, but oxygen normally exists as O2, so that's why we write half, and we're going to take 2 H+, those are going to come together from those, and then from using those electrons from the complex 4, we are going to make H2O. And so our O2 is our final electron acceptor. Okay, so that's the first part of this process, and that's the electron transport process. So at this point, we have taken that NADH and that FADH2 that we made in stages 1 through 3. We have oxidized all of those. We have taken their electrons. Their electrons are shuffled through the electron transport chain, and that shuffling of the electrons, we use that energy to then pump protons into the inner membrane space. And then when the electrons get to complex 4, we then work with the final electron acceptor, which is oxygen, and we make water. And so we call aerobic respiration aerobic or aero because you need oxygen as a final electron acceptor. So now let's focus on the right side of our figure. While all this has been happening, we have been pumping a lot of protons. And so now we're ready for the chemiosmosis part of this stage. So what we end up with is a high concentration of protons in the inner membrane space and a low concentration of protons in the matrix. And so one thing that we haven't labeled yet is this tiny little piece on our ATP synthase. This is the proton channel. And as the name proton channel implies, you can probably guess that the protons are going to flow down through that proton channel. I apologize. I don't know why I want to keep calling it a protein channel. And as those protons flow through the proton channel, what that's going to do is turn ATP synthase. As ATP synthase gets turned, we are taking ADP plus P, and we are making ATP. So again, as these hydrogen ions, these protons, as they flow down the concentration gradient, they provide the energy to rotate ATP synthase, which is a motor, to make ATP. All right, so let's go ahead, and what I would like to do now is I want to show you a quick video, and the video focuses on ATP synthase. So it doesn't walk us through the electron transport chain, but it shows us how that ATP synthase moves. So we're not going to play the whole thing. I just want to show you enough so you get an idea of how that motor works. And then I will go ahead and post the link to this video in the description box as well.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP.
Okay, so I'm going to go ahead and pause it there. Again, this video gives us a little bit more detail than we need to know about ATP synthase, but I like it because it does a really nice job of showing us how these protons go through the proton channel, and then how this ATP synthase actually gets turned to form the ATP. So again, what I encourage you to do is to walk yourself through the electron transport chain in chemiosmosis. So go ahead and draw a membrane with complexes 1 through 5, and then see if you can draw all of the pieces and see if you can recreate this model that we drew together. And then after we talk about anaerobic respiration and fermentation and photosynthesis, we're actually going to do a modeling activity. So we haven't done one since our protein modeling activity, but we're going to do a modeling activity that is based upon aerobic respiration. And so we will revisit these ideas and you will have more practice with this. Okay, so let's go ahead and do a summary of our oxidative phosphorylation. So let's go ahead and first, let's remind ourselves of how many NADH and FADH2 from stages 1 through 3 there are. So we have 10 NADH and 2 FADH2s. And again, this is from stages 1 through 3. So if we go back to our diagram here, we drew most of them coming from the citric acid cycle because most of them come from there. But keep in mind that the ones from stages 1 and 2 are going to be brought into the mitochondria, and they're also going to be used by complexes 1 and complexes 2. So this is important because we can actually figure out the total number ATP that we can get from each of these. So both NADH and FADH2 get oxidized. So when NADH gets oxidized, so this is by that complex 1, we can actually get up to 3 ATP per molecule of NADH. When FADH2 gets oxidized, we can get 2 ATP per molecule. And again, that's getting oxidized by complex 2. So we get a little less ATP from FADH2 than NADH. And part of the reason for that is it enters the electron transport chain a little further down than NADH does. So NADH has a little bit more potential energy than FADH2 does. So let's go ahead and use that and do some math. And let's calculate how many ATP we could get. So if we have 10 NADH times 3 ATP per molecule, that could be about 30 ATP. We have 2 FADH2 times 2 ATP per molecule. That's 4 ATP. And so this is 34 ATP, and this is at the end of stage 4, but keep in mind that from stages 1 through 3, we also have 4 ATP. So if we combine this 4 ATP and this 34 ATP, we could get a maximum of 38 ATP. Realistically, it's more like 36 to 38 ATP. And again, that 38, this is the absolute max. So you might be wondering, well, if we get 4 ATP from stages 1 through 3 and then 34 ATP from stage 4, then why did I write 36 and not just 38? So if we think about this, we know that when we make NADH in glycolysis, that happens in the cytosol. We then have to take that NADH, it has to be brought into the mitochondria so that complex 1 can go ahead and oxidize it. That actually takes a little bit of energy. So because of that, you sometimes get less than the absolute maximum. All right, so just like we have been doing for all of our other stages, let's go ahead and summarize stage 4. So for the location for a eukaryotic cell, this is in the inner mitochondrial matrix. For a bacteria, this is in the plasma membrane. So what goes in? Oxygen goes in as our final electron acceptor. NADH and FADH2, and this is from stages 1 through 3. And what comes out, we get the formation of water when oxygen accepts those electrons with the 2 protons to make the water, and we also get lots of ATP. So in general, what we do is we couple electron transport to ATP synthesis. Okay, so we've got a couple more things to do. One of the things that we're going to do is we're going to make a chart that summarizes the location for bacteria and for eukaryotes. So let's go ahead and we're going to do stage. And then we'll do eukaryotes versus bacteria. So go ahead and pause the video and see if you can fill this table out. All right, so stage 1, glycolysis. Regardless of whether you are a eukaryote or a bacterial cell, this is going to happen in the cytosol. Stage 2, which is the formation of acetyl-CoA in eukaryotes, this happens in the matrix. In bacteria, we are still in the cytosol. Stage 3, which is the citric acid cycle, occurs in the matrix of the mitochondria for eukaryotes. For bacteria, still living life, and it is in the cytosol. And then last but not least, for stage 4, for electron transport and chemiosmosis, this occurs in the inner mitochondrial membrane. And for bacteria, this occurs in the plasma membrane. So again, just a nice little summary chart for us to keep track of where everything is happening. So we've got 2 practice problems that we are going to talk about, and then this will be the end of aerobic respiration. So the first practice problem that we're going to talk about is thinking about what would happen if the cell ran out of oxygen. So go ahead and pause the video and try to work through this practice problem by looking at your drawing of the electron transport chain. All right, so if we take a look at our electron transport chain that we just drew, and if we find where oxygen plays a role near the end of the electron transport chain. So oxygen is our final electron acceptor. So if we have no oxygen, then we would have no final electron acceptor. So that is true. For B, B states that water would not be formed. Water is formed because oxygen is the final electron acceptor, and that combines with 2 protons. So that is also true. And then C, the electron transport chain would stop. Absolutely. If we have nowhere to dump those electrons once we're done harnessing the energy from them to pump protons, then we're going to stop the electron transport chain. It's going to cause a huge backup, which means that not only will we stop the electron transport chain, but we also would have no ATP that would be produced. And so all of the above are actually true for this practice problem. So let me know if you got this one correct. All right, so one more question that we're going to talk about, and it's an application question. So how many of you have ever heard of cyanide poisoning? So I'm sure in many of the classes that you have taken, depending on which humanities courses you've taken, that there have been discussions of people that have been poisoned by cyanide. And so the question is, well, how does cyanide poisoning work? So cyanide poisoning is an inhibitor of complex 4, and the cyanide ion is a Cn-. And so because it's an inhibitor of complex 4, we say that it is an enzyme inhibitor. And again, see if you can use your drawing of the electron transport chain to predict what would happen if someone was poisoned. So much like our previous practice problem, this is affecting the same complex. Rather than affecting the complex by not having a final electron acceptor, we have a final electron acceptor, so you can still have oxygen, but complex 4 is broken, which means that enzyme activity is now stopped, which means it cannot pass along those electrons to the final electron acceptor. So just like in our previous practice problem, everything gets backed up. So we stop the electron transport chain, we can't synthesize any ATP, and so the way that cyanide poisoning works is by breaking that enzyme. And just so you're aware of some of the symptoms that patients get, they get things like a headache, dizziness, weakness. Eventually this leads to low blood pressure, a slow heart rate, and then eventually, if exposed to enough cyanide, the patient will actually pass away. Typically, it's from respiratory or heart failure. So essentially what cyanide does is it inhibits that enzyme, and it stops your body from being able to harness energy. It stops the electron transport chain. It stops chemiosmosis. Chemiosmosis is at first dependent on the electron transport chain, and so slowly the patient ends up dying. So hopefully you are still with me and I haven't lost you. That actually ties up all of aerobic respiration. So next time what we are going to talk about is we're going to talk about anaerobic respiration. So respiration in the absence of oxygen, and then we're also going to talk about fermentation. So thanks for hanging out with me. I hope that you all have a wonderful rest of your day, and I will see you in the next video. Thanks everyone. Have a great one.